First, I would like to thank LabKey for the invitation to speak here, so we can share our experience with all the users here about the project we did since last year to manage the proteomic data for our discovery programs. This is really a team effort, so you can see quite a few people listed in the authors right here. One of them, Jonathan, is right here in the audience. For the talks, I will try to give you a justification why we need to manage data and what kind of data we have, and then focus on the customization we do, modules that we built with help from LabKey, and then also dive into the UI customization and what we think we should look at the results. And I probably focus less on the details like how we model the data, but hopefully talk about the lesson we learned along the way. And if you have any question, you can stop me anytime. Maybe you can't wait till the end, but hopefully we'll have a good discussion. So like I said, we belong to the discovery part of Medimmune, which is a part of a larger company called AstraZeneca. So our mandate is to discover new drugs. So what I think what we are trying to move into, and slowly, is to a way that we can do a data-driven discovery. It's not so much as the initial, like what we talk about system biologies and profile everything, but we want to move sort of past the dissolution phase into a more productive phase where we actually want to have focused experimental design. We want to do a lot of the molecular profiling. And so we can understand the relationship between the genome type, which comes from genomic efforts, genetics, but we want to measure a lot of the protein and peptides and et cetera to understand what these, how these entities actually drive the target, the biomarkers, and et cetera. So when I look at this approach, I can see there are some very critical components that will be necessary in order to do this one. One would be, yes, we want to be able to manage the data because we generate so much of it. But we also want to have the analytics in the visual. So basically, we want to have the data, we want to be able to understand the data, then we also want to be able to use the data to predict or to generate some decision from that point on. And I think we're going to focus on this part of that, and because we're still very far away from anything useful. The platform that we're going to focus on is going to be the proteomic, and it's a little bit different from Panorama modules that some of you are already using it. Here we try to do a discovery type of the proteomic where our goal is to quantify as much protein as possible from a given system. So with this, the new instrument we have now, with some of the novel chemistry where we can barcode the samples, in this case we talk about TMT labeling experiment, we have routinely achieved a very good coverage of the biological systems. For the cell line-based system, you are looking at the 8,000, somewhere over there. So that's practically approaching full proteomes. We're doing much less in the plasma because of the dynamic range and all the other problems. So we can start to provide the quantitative information about the proteins and then help us to integrate that information with genomics, with the transcriptomic data, and be able to answer some biological problem. But as people who have done the proteomic knows that these all generate a large amount of data, probably not in the order the same as genomic data, but still, we have a lot of data. But very importantly, it's very complicated data. That means that we cannot just basically writing down the protein conjugation at the end, and we still need to actually drill down into the raw data from time to time, and also walking back through the step where we derive the data and understand what data means. So that really just requires that we have a system that allows us to look at different aspects of the data acquisitions. I think this slide is pretty straightforward for everybody here, that this is the light pole from 2 years ago in the SMS. And I like this one because everybody complained about they don't have these tools, so that's why we need LabKey. So the way that we have been looking at the data, so in the lab is to, well, we have the study, going to acquire a bunch of raw files, and we go through a vendor supplied software, and then we have a bunch of Excel spreadsheets, and then you flip through that and try to make some sense out of that. And so the LabKey is sort of a system where we try to do a little better to fill in this blank right here. So what we try to do is try to say, okay, what is the best way to take the data we have acquired? We have spent so much effort to acquire these data, and how do we make sense of them? And how do we manage the data? And so we spend quite a bit of effort to find the system we want. And I'm not going to walk through each of the steps, but you can see it's a multi-step process. We want to go from the data to the biological interpretation. So at this point, as typical with many of the pharma exercises, that we go out to look for a solution. Say, how do we implement that workflow? Again, we could go with, in this case, I use the analogies of house hunting. You go look for a house. You could go for a fully furnished estate, which would be a typical commercial system, with a building analysis tool. And so you put data in, do all your analysis in that system, and you look at the result in that system as well. So we actually did evaluation with one of these systems, and I had to say that it looks great during the demos, but when we put our own data into it, it defined the workflow we wanted, we couldn't even finish the analysis. So we kind of give up in that approach. The other approach that I think we're also kind of familiar with it, and I have done that myself in the past, is to write something on your own, like starting from a vacant lot. Build anything you want, right? That takes a lot of time. And also the problem is it's very hard to maintain it. And it's just not a very good way to go forward. What we decide to do at that point, at the point when we fail, so for that initial evaluation, is say, okay, we need to break down these overall workflow into different parts where the tools that do the best for the part should still do that. Like, for example, the database searching and the protein identification, we're going to do that in ProteomeDiscoverer or Maxquant, the tools that people generally use them. And then we're going to do the analysis in R. We're on R script, analyze the data, doing the stats. But we're going to use LabKey to manage the result of analysis and help us to organize the data. And then for the part that visualization and further analysis that we could not do well in the LabKey, and we can still do it in R Shiny. So that's our solution for the in-depth analysis. So the model, I think, works out in this case is that in the areas of open source, where we have a lot of the tools in the open source community, that instead of either having fully built a house or a vacant lot, you can start with something that has a skeleton, has an infrastructure support that you can use. Then you also have access to a lot of the widgets, a lot of tools, like you have Home Depot free for you. You have IKEA free at your disposal. So what do you want to do to build your house? You can either do it yourself or have the help from a professional, like folks in the LabKey, who were like designers and builders who help out to build the whole things. So it still takes a lot of efforts to go from this point to sort of become a livable house, but it's much better than starting from a vacant lot. The first thing I would describe is how we're going to import a study. So for the people who have not done the proteomic work, it really consists of, it's like any platform, omic platform, where you're doing a study and have a bunch of samples, and you're going to run your instrument, and you're going to generate a result. and you run through some kind of the tools to interpret them. In this case, we start an investigation, and that could have different set of the study, means that you're going to run one set of the mass spec run, and maybe a different set of mass spec run for particular projects. And then each one of them consists of many samples, sample set, all kind of metadata. So when we are looking at the LabKey solution, so it become apparent that we kind of need to model this relationship that describe our study and samples. And we try to look at the LabKey schemas, and there is a proteomic component in our LabKey, but we felt that, at least for our own data, it's a little bit easier to just to come up with a new set of the table to describe these kind of relationship. One of the things that we had to learn sort of way around is, in the LabKey, I'm sure you're aware of that, everything is sort of organized by folder, right? You set up your folder, that's the scope for the access control, and you put your files there and decide to do something about it. In this case, that's how we organize the data as well, that we have top-level projects, LabKey projects, but we also define the folder that can either be investigation study or samples or analysis sitting in either a single folders or multi-level folders. And then we design a way, sort of a logic, say, okay, if you take the output from the ProteomeDiscoverer that already have the sample relationship already built, and we have the protein ID, we have the peptide ID, have the protein intensity, etc. We, with our user a team together, we design a set of the logic, say, okay, how do we parse these data files and then load into the table that we set up inside the LabKey? So this is what we did, and with the help, then we passed this set of the table design and logics to Frank and Corey's and all nice people in the LabKey, and they actually implemented a module called Medimmune module for us that allows us to import the data. So I'm not going to go through how we organize the mass spec related data, except that this particular diagram was try to model what we call TMT labeled quantitation data. So isotopic labeled proteins. We also want to extend this set of the table to label free or even just simply qualitative data. But in essence, that this will sort of encapsulate a typical discovery proteomic data type. And then following the data loading, we will also have a dedicated efforts to do the statistical testing. Basically, you run the protein quantitation, but then you really want to find out which protein has changed significantly from one condition to the other. We have our statistician that set up these models and do all this testing. So at this moment, we're doing the stat outside of LabKey because we have not feel comfortable that we can actually have a candid pipeline to do that. So we actually wait. So once we have data import, we can then click on the button right here and to load the statistical outcome back in the database and then merge these two data together so you have a complete data from metadata to the protein peptide ID to the list of the fold change, p-value, etc. So, so those are all the sort of implementation logics, right? From the user point of view, what you're going to see in our instance of LabKey is that you will go in and you will create a necessary subfolder. You upload the data that are coming out of the ProteomeDiscoverer or maybe in the future in a Maxquant. And then you can say, I'm going to create a folder that is of type Medimmune in this case. That invoke the Medimmune modules. But a better solution that what we find out is that with help from Steve and other people doing the training is that if we want to incorporate a set of UI customization, it's actually best to create a template folder where we not only have the module assigned to it, but also have set of UI customization already done to that module, to that folder. And then when you select create from folder, template photos, you automatically inherit that set of customization, which I will show you in the next few minutes. So I think this is a reasonable way to sort of bring a set of the UI that we think works best for our data with that module for our own studies. In terms of the data loading, it's actually quite straightforward. That once you load this data, so people who have used ProteomeDiscoverer will recognize these are the files that you export from the vendor software. And then you put them in the folders, and then you click import data, and you go through a form that's asking you various parameters you're going to feed into the import. And one of the important set of parameters is how you're going to map what ProteomeDiscoverer calls something into a control vocabulary, like protein sequence or peptide sequence. How are you going to do that? And then once you fill out the forms, then click on submit. We have a set of R script we implement, we call it a pipe script, where LabKey will call the R script at particular entrance points, and then pass on the files that we have submitted. And at the endpoint, the pipeline will pick up the parsed files, and then the data loading logics from LabKey Medimmune modules will then load those data into the table that we have designed. So that will complete the task for the import data, and once this is done, the data is loaded, right? So that will complete this particular task. But then for the actual scientists, it's not really big helpful, because you load the data, if they still don't have a way to look at the data, it's not quite useful. So we put a lot of effort trying to do, okay, how do we visualize data? How do you let people browse through the data? And so I'm going to spend a few minutes to talk about that. So as everybody probably already know that I think the best feature of the LabKey, besides the infrastructure it provided, like user control, access control, servlet, all of the infrastructure, is the way you can customize the UI to like infinitesimal possibilities. In this case, what we did is we took a typical layout, I think this is from one of the clinical study portal, something like that, where you have a multi-tab layout. So this is fairly typical. And then we first customized the tab that we want. We added tabs or hide the tabs or whatever we need to do. And then once you have the tab, this one thing that you could do is called select web parts. And there's probably a lot of choice you can go through, but we use the one called query web part, which basically allow you to put the data grids on the web page that can be backed by the SQL query that we can write against the table. And that really gives us a lot of the possible ways to slice and dice our data. So that's really, really cool. So the tab we have an overview tab showing what the data we get loaded, and the import tab is the one that functionality I reviewed, where we put the data in and do the import. We then have the ability to review at the sample level, like what's the protein detection, quantitation for each of the samples. We can look at individual run that come through from our experiments. And then importantly, we can look at the protein levels, and that allows us to look at a peptide level and possibly spectral level as well. Then we have a lot of other grid that we don't know what to do with it. We just put it into about, like how do you edit each of the table. But for most of the time, people will be dealing with these first few tabs, allow them to look at the result of the study. So I'm going to flip through a few of the screen. So the first one actually is to show you, for the people, for ones who have not tried, that it's pretty straightforward to do that. So when you click on the select web part and select the query, you will then fill out this form in which that you select the modules and the query that you want to produce the data. most of the time, we'll write a query ourselves, and that becomes the customized query when you look at the listing, and which is just ordinary query with a little bit of the LabKey sort of requirements. And mostly it's to do with the container scoping and how the user access control is applied. But this is really nothing more than just a SQL query. You write it, and then the query will produce a data table, such as the one that we listed here. Another very powerful feature that we learned is that you can put some link behind the number. So in the customization that you specify right here, and probably a different screen right here, with this very complicated arcane syntax, you can program the link so that allow people to click on these blue links that will pop it out to something else. And that's very useful because that allows us to extend the LabKey further into something else that's probably not best suitable for itself. So this is for people who are doing discovery proteomics. This will become apparent that this is a way to drill down from the protein down to peptide to PSM and looking through all the information that you ever want to know. But one thing we don't have in the LabKey system is we do not store the raw file for practical reason because it's too big. And most of the time, we don't really need to look at the raw files. But I will show you one way to access that. But I'm not going to go through all these detail right here because those are not interest for people who really don't do it. Just to show you that this way to customize the UI that allow us to do this. We also learned that the report, especially our report, is fantastic because we used to do these kind of bar chart, box plot, or this plot with Shiny, where we need to take the data frame out and do Shiny. But with a very few lines of code, you can take exactly the same code in R Shiny and plug into the LabKey.data and basically produce the same kind of plot right in your LabKey system. So we see that, okay, my favorite proteins has the change in the abundance from the baseline to a different condition. People really love to see that because they have their favorite genes, favorite proteins. They can go in and see what do we actually measure. And if you really, really want to know, you can see at the peptide level or spectrum levels how the abundance change from condition to condition. So that better to be consistent with the protein information. But sometimes it's not so. So this is a great feature. But I think the only one we couldn't really get to work with this feature is that the definition of the cohort is something that we have to basically hard-code it for each different study. We have difficulty making a generic R report that can pick up the experimental definition and automatically lay out the plot that we want to be. So that is still a little bit difficult. But because we only do one study every few weeks, so it's okay to do it by hand. So this is the point where we want to extend the LabKey to the point where if I want to see the MS spectrums of my favorite peptides from this run, but LabKey doesn't have that because we did not import the raw ion information into our system. So we took advantage of another system called OpenSlice. This is open source, which is a set of Python script that pre-process your raw files into some intermediate, actually it's MySQL database that's hosted in separate machine. But you access them through a custom URL right here for the peptides and through the, right here, you program the URL, so when you click that, it'll pop up a spectrum for you. And you can also look at the TIC trace and select the XIC trace. So that we can actually achieve the access to the raw data by extension to a link to another piece of open source programs. Another thing that we have to do from time to time is that although LabKey is extremely powerful, but I think most of the time it's really good for plotting and charting and take your data and plot a different way. What we find that is for something that's more extensive, like if you want to have very customizable live tables or volcano plot that actually when you highlight the points right here and pop up the other way, kind of linked volcano plots, or in enrichment analysis, heat map, or sort of other things that bioinformatics people like to do, it's actually easier to do it in RShiny, much easier to do RShiny. So we actually have a separate RShiny app that took the same data that LabKey is working with it and basically work off that and allow people to do a much more extensive analysis. And then we can build the links to that Shiny in the wikis or there's part of the UI so people to jump into that if they need to see more information. So that's one thing, and I think that lesson is that I think we want to use LabKey to manage the data, but because it's open source and very customized for architectures, we can actually link to very different part of the system. But I have to say that in order to really use the system well, I think the lesson, or at least the architecture design that we kind of learn along the way is that you probably, at least what works for us, is to isolate the part of the analysis that's sort of platform specific to your own script or program or something else, because LabKey can tap into that pretty easily. So in our case, what LabKey serves is a data management tool that we have data coming in, we're going to do a next step, and do a next step, and we capture the results. But each of the analysis steps is all done in the script that we have control over with it. And then, like I mentioned, the UI customization we do really works better if we ask people to always create folders from the templates. And then, so the things that we learned, other thing we learned is that I mentioned already that the folder concept is extremely powerful. It's really necessary to manage the access of the data, but in a way that it could also contradict a little bit to how we want to organize investigation, study, and analysis. So we really have to learn the system well, and I have to say that it took us a while to learn what is the LabKey speaking kind of things, how LabKey organizes the data. We still kind of sometimes struggle with how the folder is organized versus how the data logically should be organized. We're still learning that. And in terms of implementation, I think the best way for us is to design the system, design the requirements, but then ask LabKey folks to do the implementation if they touch the server side part of the code. If it's UI part of the code, we should try our best to do the customization ourselves, because that involves too much of the conversation that, what we want this way, that way, and then sometimes it's just easier to write your own custom queries and do the R reports and do those kind of things. But still, if you need to design a different set of tables, you need to load the data, you need to design a form that will import taking the parameter, it's certainly best be done by the professional. And the last thing is I kind of hesitate to say that because when the first time we evaluated commercial products, everybody got blown away by all these nice plots and things that you see, although they didn't work out for us. But when you look at a LabKey, at least in our instance, people are just somewhat seeing the lacking of those kind of things. But I think that, so it took quite a bit of work and sort of imagination to see what LabKey can do from that point on to convince our user that yes, if you actually do it and you actually design in the UI customization in a way, this is what we're going to see. Because when we first demoed it, it's a little bit different from what we were expecting it. But this is nobody's fault, but this is the way that I think it's the advantage of LabKey to the point where it gives you a very nice infrastructure and skeletons and a lot of customization, but you still have to learn the system very well. And we're still doing the phase 2 with LabKey that really involved basically be able to look at a label-free quantitation and possibly other data type. And we also want to optimize the import UI because the first version we did was quite cumbersome, and we are in the process of optimizing that. Certainly, we're going to implement basically the panoramic modules because we're ramping up those kind of activities. So we all continue to work and basically enhance the system for our own needed. Certainly, I would like to acknowledge that all my colleagues from different departments in our company, research mathematics, with ITs, with protein science, who actually do the experiments, with our statistical people who do the statistical modeling. And for the people we work with from LabKey, everybody's really, really superb and really, really good and give us a lot of help. So, and again, thanks for your time and